Welcome everyone. Thank you so much for joining our webinar today. My name is Susan Chung. I am the Senior Global Product Manager for Quantabio. We have an exciting talk for you today on the topic of cDNA synthesis using Quantabio's QScript Ultra kits that are inhibitor tolerant and enables rapid full-length synthesis of cDNA even at elevated temperatures. All right, just a quick introduction on who we are and what we do. Quantabio is an innovative life sciences brand. We were founded in 2001 by amplification scientists from LifeTech. We are looking to develop products for RT, qPCR, PCR, and NGS that combines application-specific enzymes with optimized reagent compositions with the ultimate goal of combining these two aspects for superior product performance. And with that, I'll introduce to you all our speakers for today. We have two amazing speakers from our Quantabio team. First, we have Dr. Ryan Heller. He is our principal scientist for product development. He's been with Quanta for almost a decade and has been vital to the development of some of the amazing products that are in our customers' hands today. He received his PhD from Cornell University, specializing in DNA replication, recombination, and repair. And before joining our Quanta team, he was a part of GE Global Research. And next we would have Dr. Subrata Panja. He is our Associate Director of Field Application. He joined the Quanta team about 4 years ago, and he brings about 15 years of research experience prior to contributing his knowledge to field applications. And one of his previous work was at GeneDx, where he worked on diagnostic assay development using NGS, qPCR, and dPCR. Just a few housekeeping items before we delve into the talk. Please keep your microphones muted during the webinar. And for your questions, please use the Q&A function within Zoom. They will be addressed at the end of the webinar. And the webinar is being recorded, and the recording will be available after the webinar. And with that, I'll hand it off to Dr. Heller.
Well, thank you, Susan. Thanks everybody for joining. So yeah, let's talk about cDNA synthesis and some of the new products we have and a new enzyme we have. But first, I'll just get started by talking about what is cDNA synthesis and why it's important. So in concept, it's relatively simple, right? So we're starting with an RNA template, and the idea is to convert that into a cDNA product for which we can use for some sort of analysis, right? And so to make the cDNA product, you need a reverse transcription step. And then analysis is typically done by PCR. And so relatively simple, but important. And the details are important because this method, though simple, really underpins many methods for RNA analysis, including gene expression quantification, non-coding RNA profiling, RNA pathogen quantification and sequencing, whole transcriptome and transcriptome sequencing, and then analyzing transcript variants. So what are the details in this process that we need to think about, right? So first is that for really quality results, we need that the RNA sequence is converted accurately and completely into cDNA without bias. And this bias can happen through GC content or length or sequence. So really, if your system can produce a cDNA that's unbiased, this is ideal. High efficiency conversion, and this relates to the kind of the levels of RNA in the template, right? So we want to have equal conversion of kind of high abundance products and low abundance products. And the low abundance products can be either low copy gene expression products or single cell samples. So really, we want to maintain consistency over a broad input range. Length of cDNA product, for some applications, this is important. Maintaining full length sequence analysis of these transcripts can be challenging, right? So RNA is known to have secondary structure. Oftentimes the reverse transcriptase pauses or stalls, and you can get products that are truncated. So a system that can produce long cDNA products consistently can be a big advantage. And finally, this is maybe kind of less known or less considered, but compatibility with amplification reagents. So, because it's by definition a two-step process, reverse transcription, cDNA synthesis, and then analysis, there'll be some carryover of the first strand product, into your analysis step. And what's coming from that first strand product can impact how it's, how the analysis takes place and can impact the results there. So this is something to think about, and I'll get into this further later in the talk, about what's carrying over and how to deal with that. So choice of RT system, reverse transcriptase system is critical. There's many factors here to think about, and I'll go through these each sort of individually in the upcoming slides, so I won't mention that in too detail now. Okay, so for cDNA synthesis, a reverse transcriptase is the critical enzyme there. So reverse transcriptase, by definition, it's a magnesium-dependent, RNA-dependent DNA polymerase. So you're producing cDNA or DNA from an RNA template. And most commonly, the RTs that you'll run into commercially are derived from a retroviral source, typically from one of these three categories that I have listed here. So one possibility is an RT from an HIV or an SIV type source. So these are two subunit enzymes, heterodimers, and typically these have high intrinsic RNase H activity, with the RNase H subunit being present on one of the subunits of the heterodimer. And these do not template switch well. Some types of RNA analysis require a template switching in which the reverse transcriptase will leave a few nucleotides at the end of your product. But for this particular class of enzymes, because that sequence of non-templated nucleotides that are left there are less predictable, sort of more random, standard template switch oligos don't tend to work well for this class of enzyme. The second one I have listed here is the MMLV class. This is probably the most common RT that we'll run into with commercial enzymes, commercial modern RT enzymes. It's a single subunit enzyme, relatively easier to produce. It has moderate RNase H activity. And as I said, most RTs that you'll run into are this class. And finally, the AMV class, also a two subunit heterodimer with high RNase activity, with that RNase H domain present on both subunits. So I mentioned RNase H activity, and this is something that's evolved to work well with, reverse transcriptases in nature. But as a molecular biology tool, sometimes this can get in the way and cause issues. So by definition, RNase H is a activity that will cleave or degrade RNA and an RNA DNA duplex. So you can imagine, if your template is composed of RNA, which it would be in this case, having an activity that's going to degrade this might not be ideal. So oftentimes this RNase H activity will be eliminated or reduced by mutagenesis. And some of the improved properties that come from these RTs with the reduced RNase H activity are higher first strand yield. Because you're not degrading your RNA template, you tend to get more product this way. And then higher sensitivity, because you're not degrading those low abundance transcripts and stalling and losing that quite as frequently. Greater percentage of full-length cDNA, again, reducing that RNase H activity kind of helps the RT from stalling out and losing that RNA template as you're generating long product. So getting rid of that activity, you tend to get better processivity, get more complete representation and full-length cDNAs out of it. So this is obviously very important for these sort of analyses. Maintains template strand specificity in first strand product. I mean, for some sequencing applications, this is important. Again, you're sort of maintaining contact with the strand of RNA that you're interested in. You can sort of get less promiscuous activity and jumping to different strands. So you get more accurate sequence representation in the final product. And then finally, it's been found that actually mutagenizing or reducing that RNase H activity in RTs tends to lead to a more kind of stable enzyme structure, which improves thermostability of the enzyme and lets you run reverse transcriptase reactions at higher temperatures. And that high temperature cDNA synthesis is helpful in many cases because RNA, as we know, has a high degree of secondary structure often. And that high temperature can help relax that, allow your primer to get better access to your template, and just allow, more robust RT in general. So these are some advantages, and most modern RTs now are reduced or eliminated at RNase-H. But there is a drawback that should be considered, and that is, as you've now generated your first strand product, because that RNase-H activity is no longer present, that RNA template's still around, right? So you haven't gotten rid of it. And so in PCR analysis, sometimes this can get in the way, partially dependent on the sequence composition of that RNA and the length of it. During PCR denaturation, if it happens to be that long duplex now doesn't fully separate during the annealing step, it can renature. And if that takes place, now you don't have access quite as well from your DNA primers to your cDNA, and attack can't get access to that DNA primer anymore. And so your PCR efficiency now is greatly reduced. So it's something to think about and keep in mind when choosing an RT system is how that leftover RNA, that carryover RNA is dealt with in reactions. Okay, so next I'm going to get into one of our new engineered RTs that we've called QScript Ultra. And so this is a non-MMLV based enzyme that we've produced in-house through a series of mutagenesis and variant screening. And we focused on three major domains of the protein, one being that RNase-H, reducing that for the reasons I described earlier for improved properties, but also some other regions that are kind of known or predicted to interact with the primer template nucleic acid material, such as the minor groove binding tract and the connection domain. So increased affinity for that primer template leads to some improved properties that I'll go through here. So one being the thermostability. We know that's important because of RNA secondary structure. So if you can run reactions at higher temperatures, you can improve your cDNA yields. Processivity. Here we're defining it as the kind of propensity of your RT to remain engaged with the primer template during elongation. So less occurrences, less frequency of falling off, less frequency of stalling. And what this leads to in reactions is a better generation of long cDNA products, again, less premature stalling, and a faster reaction because you're not having that same degree of the enzyme falling off and having to kind of re-engage with the primer template. This can lead to shorter reaction times, faster time to result in your cDNA synthesis reactions. And again, so robustness is another kind of category that we've been able to improve with this mutagenesis program, where better affinity for the primer template, better ability to engage means that your inhibitors are less effective at removing it and disengaging it. So you get better tolerance to salt and other common reaction inhibitors, and these all could go into a little bit later. Yeah, and then finally, reduced RNase-H activity, which, as we discussed, leads to higher yield and more full-length products as well. So one question that comes up is about fidelity of reverse transcriptases and if this one is changed at all from common RTs. So we went through this published protocol in which we looked at how the RT mutation rate can be quantified through cDNA synthesis. And so what we did was take an input RNA and then use a bar-coded primer for that first strand synthesis. And that lets you go back later on and then kind of associate each individual cDNA synthesis product with a particular barcode. And so after library amplification, you can actually bin those barcodes and then sort out errors that would have occurred from the sequencing itself from those errors that actually occurred during cDNA synthesis. So eliminating anything where you'd see it, occurring just once in the set, and then, where it's consistent, we know that occurred from the cDNA synthesis, and those are legitimate RT errors. And so what we found, interestingly, was a relatively low frequency of insertions and deletions. Out of the quantity of bases that were analyzed, actually 0 insertions and only 14 deletions were found. In this case, 166 mismatches. So just getting to the mismatch rate in general, we know that RTs are, because they're not proofreading enzymes, they're sort of, known to be kind of mutation prone or have less fidelity than common, polymerases that have the proofreading activity. So we found, in this case, one error out of every 27,000 bases, which fits within the range of published error rates. So no really difference in that between this RT and other common ones. But the one interesting thing was the deletion. So the insertion deletion rate we found to be quite substantially different than a standard MML VRT. And we think this actually reduced indel rates, maybe related to that improved processivity. So you can imagine if the enzyme itself has a less frequency of stalling or slipping or this sort of thing, that it might lead to less insertions and deletions. So we're exploring this a bit further, but this could have interesting implications for various sequencing protocols. So having this sort of new evolved generation of RT, our goal was to next incorporate this into a complete system for cDNA synthesis. In this case, we've called the kit the QScript Ultra Flex kit. And this is meant for, cDNA synthesis, but with flexibility in how the material's primed, right? So it would allow for oligo DT priming, random priming, gene-specific priming, or combinations of these various strategies. Depending on your material and your priming strategy, compatibility can be with the total RNA or with mRNA containing poly A material at the 3' end. And it's meant to be compatible with real-time RT-qPCR, endpoint methods, and sequencing as well. Okay, so why would you want a kit that's got flexible priming methods? What are the different priming methods, and how does each one, have advantages and disadvantages? So one method is to use gene-specific primers. In this case, user would provide their own primers. This is, by definition, a more specific method than a general cDNA synthesis. One disadvantage is maybe in your choice of primer, because if you were to happen to choose a region that's located within a region of secondary structure, it's possible that that secondary structure could have an impact on your product yield based on how much secondary structure is present there at that region. A second method that we would allow, we would provide a tube for oligo DT priming. Because this oligo DT is priming now on the poly A tail of mRNAs, it's your template's limited to only those products that have the poly A tail, so those mRNAs. And this is quite effective at capturing sequence from the 3' end. And one known disadvantage is because you're starting at the 3' end, that it requires that you sort of go through and do cDNA synthesis on the entire molecule. What can happen with this method is that you'd see potentially underrepresentation at that 5' end. And then a third method is random priming. So an advantage of random primers, they're smaller, they prime on any RNA template, no sequence requirement at all. And these are great for capturing regions of RNA at the 5' end. But because of how they're hybridizing to RNA, what can happen is that your 3' end can be underrepresented. Okay, so the kit itself, we've got some specifications here. We've been able to provide the buffer and enzyme in a master mix. We've got separate tubes of primers. It's a 5x concentration. And one really unique feature, I think, of this kit, because of the improved processivity of that RT, is that you can generate really long amplicon lengths out of this kit. So up to 20 kb, in just a 10-minute reaction time. The setup of reactions is pretty straightforward. Classically, one would do this two-phase setup. where you would incubate your primer mix and your input DNA, separately and set that up initially, and then heat that at 65 degrees for 5 minutes. The idea there being that this would help relax the secondary structure in the RNA, allow your primers to anneal, and then place that on ice, add your reaction mix to start the reactions, and then go ahead with the cDNA synthesis. Now, what we found is because the RT itself has that higher thermostability, and we're able to do reactions at elevated temperatures, in this case 55 degrees, whereas typically, unevolved or unmodified RTs are done, classically at 42 degrees or so. This increase in the temperature kind of helps you bypass this step. So really, in most cases, we found that a simplified reaction setup is working just fine, where you can just create one master mix, 3 pipetting steps, primers, input RNA, reaction mix, and that's it. And really you can go. So really a straightforward process. So one feature of the kit is a broad dynamic range. We mentioned that this is important, and this is, through qPCR analysis, one of the big advantages of that method is that you can start with material as little as 100 femtograms, or go as high as multiple micrograms of material, and stay in the linear range. And that staying in that linear range is critical for maintaining optimal relative gene expression data. So you can imagine if you were to choose something that's off the linear range, you can be off by a few CT, and then your relative quantification based on that is going to be off by several fold. So this kit really allows a nice dynamic range. We've looked at FFP RNA as well. So this is highly degraded material. You don't quite get the same range out of it because it's degraded. But one important thing to note is that the sensitivity is quite good. So even out to really low levels of amplifiable material with this FFP RNA, still working well and compatible with the kit. So yeah, we can generate, I mentioned, over 20 kb of cDNA synthesis in 10 minutes, which is really quite remarkable with this kit. You can see previous generations of standard MMLV RT, would take 60 minutes or more, and even then have difficulty achieving this length of cDNA product. Whereas here, we're just doing a gene-specific priming of a long human SYNE1 transcript, and in just 10 minutes, you can get reasonable yield of even 20 kb product. And here we've done long-range PCR analysis with our replica hi-fi tough mix, which itself is a robust and rapid mix for generating these long products. Okay, as far as thermostability, so, in the UltraFlex kit, we're recommending 55 to 60 degrees. In this case, in this experiment, we see, how far can you really push it? So we took a stable LDHA gene-specific primer, reacted it with total RNA at different reaction temperatures from 55, 60, 65, and 70 degrees. And you can see that, even up to 60 degrees, you get robust product without any reduction in yield, whereas even some of the kind of alternative modern, modified RTs are not able to withstand that sort of temperature. Even at 65 degrees, the yield is reasonable with only, with a 1 CQ or so loss in product. So even for those challenging RNAs with secondary structure, this enzyme can work well at high temperatures, which should be able to relax those well. Okay, I mentioned earlier about carryover RNA. So again, just to remind you that one potential downside of having a RNase H minus reverse transcriptase is that you have that leftover RNA template that gets carried over to PCR reactions. And so now what we've done with this mixture and this master mix to get around that is to include, we've modified the buffers a bit, included some accessory proteins that actually, so it doesn't get rid of that RNA template strand, but it allows it to prevent it from interfering with downstream PCR analysis. So in this case, in this experiment, we did cDNA synthesis on some longer targets, generated that first strand material, and either left that untreated or gotten rid of that template with an exogenous RNase H enzyme, and then did long-range PCR analysis. So you can see comparing, this 12.5 kb product without that RNase H treatment compared to with the RNase H treatment, very little, if any, difference in cDNA yield. And it was the same for this rat brain input and the 17.7 kb product, where there was really no effect of including that additional RNase H. So it seems to be unnecessary. We haven't found a need for it. Again, these accessory proteins that we've included in the mix seem to prevent it from, causing any harm in the downstream analysis. Okay, as far as inhibitor tolerance, again, we've modified and mutagenized this enzyme to be as tolerant as possible. So this can be another issue if, you're working with blood products or plant products, or there's some carryover from extraction chemicals. This can lead to, in some instances, some delay in CQ, some inhibition, which could throw off your results and skew the data. And so having an inhibitor-tolerant, robust RT, where this is less of a problem, is definitely an advantage. So you can see here with the QScript Ultra, even high levels of heparin, hematin, just moderate effects on the downstream analysis, whereas some of the other modern RTs really struggle with that. So definitely an advantage with less pure material. Okay, so we have some additional applied work with some long-range virus sequencing that Subrata will discuss next.
Wonderful. Thank you, Ryan. So as Ryan described, that our Ultra RT can withstand high temperature, it can generate long cDNA in just 10 minutes. So we developed some application data, and in next couple of slides, I'm going to talk about an example where we utilized Ultra RT for RNA virus sequencing. So the protocol is very simple. We started with some isolated viral RNA, and then we performed long cDNA synthesis, typically around 10 kb long cDNA was our target, using QScript Ultra FlexKit. And you can imagine that we performed cDNA synthesis at 55 degrees centigrade, and that was done just in 10 minutes. And then we used our replica hi-fi tough mix, which is specifically used for long-range PCR applications. So in just 90 minutes, we were able to amplify 10 kb targets. And then once you have long PCR products, you can use those for any kind of downstream sequencing applications. If you want long range, then you can use PacBio ONT. And even you can use those 10 kb double-stranded products, fragment it and sequence it in Illumina platform. In this particular example, we used our Spark DNA Fragment Library Prep Kit to generate libraries for the Illumina platform, sequenced it, and I'll show you some data at the end. So in this particular section, we will focus on the data where we took SARS-CoV-2 RNA, which is approximately 30 kb long. Now, there are many different methods available in the market where random primers were used to generate cDNA, followed by a panel of primers for generating shorter amplicons. Now, those methods usually works well, but you can imagine for every RNA viral target, you need to put significant effort to develop those panel of primers. Here we are showing that designing primers are very simple, and you can utilize this method for 30 kb long RNA all the way down to smaller RNA with less effort, less amount of time. So in this particular example, with 30 kb long SARS-CoV-2 RNA, we designed 4 gene-specific primers. So you can see those are binding to SARS-CoV-2 RNA in the positions mentioned here. And then, as I mentioned before, we used QScript UltraFlex Kit to generate cDNA at 55 degrees centigrade just in 10 minutes. So you can get, depending on the position of the gene-specific primer, you can get different lengths of cDNA. And then we used replica hi-fi tough-mix and primers to generate amplicons of between 7 to 8.3 kb. So we ran those amplicons in a tape station, and you can see we got really nice bands of the long PCR products. So once you have these long PCR products, again, as I said before, you can use those for any kind of sequencing applications. In this particular project, we used our Spark DNA fragment library prep kit, fragmented those amplicons into shorter length ligated Illumina adapters, and generated libraries. So you can see we got really nice libraries, and then we sequenced it in MySeq. And each case, the map percentage of map reads were above 98%, and the duplication rates were low, 4% or less than 4% in most cases. And when we mapped those reads and look through IGV, you can see that we get really nice coverage across the SARS-CoV-2 genome. And in couple of cases, you can see higher number of reads, and those are the regions where you get overlapped reads coming from 2 different amplicons. And those overlapping regions are important because if there is the deletion or mutation covered by one of the primer, other primer will be able to pick up those mutations. And one of the major advantage of utilizing this kind of longer cDNA, longer amplicon-based system, if there is a larger deletion, it's very difficult to detect by shorter amplicons, and it is difficult to develop those shorter amplicon-based panels as well. Now, if you look at many other common RNA virus like HIV, HCV, or poliovirus, those RNA virus genome are less than 10 kb. So you can just use one gene-specific primer to generate entire cDNA, followed by one long-range PCR, and then you can sequence the entire RNA virus. So potentially this has a lot of different application, and different labs are getting benefited from this kind of approach. Now, I want to talk about, so Ryan mentioned that this UltraFlex kit was developed to generate long cDNA up to 20 kb just in 10 minutes. So I was curious and wanted to know, is 20 kb is the limitation of this kit, whether we can generate even longer than 20 kb? Because if I want to clone a very long cDNA or RNA virus, whether I can use this kit or not. So for this particular application, I used OC43 RNA, again, another 30 kb long coronavirus RNA. And the reason I used it because the quality of the RNA I got from this particular sample was good. So there were a lot of intact RNA. And for this particular application, I just used one gene-specific primer attached to the 3' end of the RNA virus. And then I generate cDNA at 10 minutes at 55 degree or at different time points. Here I'm only showing 10 minutes and 30 minutes time point cDNA synthesis with QScript UltraFlex kit. And then to detect whether we are getting 10 kb, 20 kb, or 30 kb long cDNA synthesis, we amplify different region of the cDNA. So for this, in this example, if we are getting this blue part amplified, that means we get 10 kb cDNA synthesis from the 3' end. If we get both blue and green amplified, that means we got 20 kb cDNA synthesized. And then if we get 3 bands, which is basically blue, green, and red amplified, that means we have a 30 kb cDNA synthesized. So when you looked at the 10-minute data, only blue and green part amplified. So in 10 minutes, we roughly get 20 kb cDNA synthesized and red was absent. But I was really excited to see that when we extended the cDNA synthesis time from 10 minutes to 30 minutes, I start getting bands on the red amplicon as well. So that clearly indicates that if we use UltraFlex kit and perform the cDNA for longer time, like 30 minutes, we can generate cDNA up to 30 kb as well. So if you want to generate a cDNA for very long mRNA or RNA fighters for cloning or sequencing application, it is definitely possible. So with that, I want to pass it back to Ryan to talk about the next product.
Thanks, Subrata. Yeah, the next kit that incorporates the QScript Ultra enzyme that I'll talk about is called QScript Ultra Supermix. So this, the formulation is quite similar to the Flex kit, except that rather than providing separate tubes for different priming methods, everything's combined into a single super mix. So this is only one tube now, which reduces the pipetting steps, allows for nice consistency and reproducibility. And because we've combined now the oligo DT primers and the random primers together into a single mix, this mixed priming strategy has some advantages that I'll talk about in a minute. And, because of that, also we're now compatible with all types of RNA, and similar improvements in reaction chemistry. So the one tube mix, as I mentioned, it's a single tube, 5x concentration. Now, because we're using random primers, and those random primers, they anneal randomly throughout the RNA template. Now, we're not generating quite as long products here, only 400 kb, but, with that comes several advantages. Yeah, so now you have the benefits of both oligo DT and random priming. I talk about this on the next slide here. So, right, if you were to use oligo DT priming exclusively, you can see here in your polyadenylated RNA template here, oftentimes the products won't reach the end, so you'll lose sequence coverage, at the other end of the material. And conversely, with random primers, you get nice representation here at the 5' end, but often missing sequence here at the 3' end. Now, combining those two, you get the best of both worlds, right? You get an ideal mixture, would provide an unbiased coverage of the RNA template in reaction product. And so we've demonstrated that with this example here with the MAP4 mRNA, where we've used this ultra super mixed priming method to generate cDNA and then analyze that cDNA at 4 different positions along a relatively long RNA template. And you can see here that the amplification curves are essentially lining up, depending on the amount of template input used for the reactions, indicating equivalent CQ at all locations and therefore unbiased coverage throughout that long transcript. So this kit works well. Again, broad dynamic range is critical. We've done some experimentation at the higher end up to 2.5 micrograms, and you can see, with twofold dilutions, maintenance of linearity. Whereas some kits, as you approach that microgram point, you start to actually lose that linearity. And this is important because if you were to be using a reference gene that's high copy, or use enough material where your reference gene would be at this level, and you're not on the linear part of the reaction, now if you're a CQ off, you're throwing off everything you've quantitated against that by twofold. If you're 2 CQ off, fourfold. So it's important as designing experiments to be sure you're in that linear range. And this kit works well in that upper range. And at the low range material, it's critical as well that you're not losing those low abundance transcripts. So here we've done a kind of a range of different transcripts from relatively high to relatively low, several replicates for each. And you can see with some kits at that lower level, you get stochastic dropout. And we found that the QScript Ultra based kits, because of the way it's formulated, the sequence and the composition and the concentration of the primers in there and the way that's been optimized, works really well at maintaining, identification of those low coverage transcripts. So in many kits, because typically your sample and your will include RNA virus at relatively low levels, these kind of column-based kits will include some poly A carrier through the purification process, which can carry over now into that sample. So your sample's now got a relatively high level of poly A and maybe only a handful of virus material, and that poly A can interfere, right? So again, this is a mixed priming method where you're using oligo DT primers. Those have the potential for sure to bind to that carrier RNA. I'll allow it to do cDNA synthesis and potentially interfere. And so we found that even with this particular kit, up to a microgram of poly A has very little effect. Whereas you can see with some other alternative kits, there's certainly some CQ delays with that poly A material, which, can lead to a loss in sensitivity. Inhibitor tolerance, we've done, we've looked at more than this, but this is some examples of a panel of inhibitors that we've looked at with the Ultra Super Mix. So just like the Ultra Flex kit, it's using the same reverse transcriptase, and because of the way it's been mutagenized and evolved, it really shows nice intrinsic inhibitor tolerance that it's providing it to these kits. Whereas some alternative RTs are really kind of inhibited or blocked completely by these levels of, different products. Okay, so I'll pass it again to Subrata for some interesting work on some 96-well gene expression panel data that he has with this kit.
Yeah, thank you, Ryan. So as Ryan mentioned that our QScript Ultra cDNA Supermix is one tube solution and generally produce robust cDNA synthesis result if you need to generate shorter cDNA. So that makes it ideal for different gene expression analysis. Now, when somebody get a new reagent in the lab, they always compare with their cDNA reagent already have in the lab and do a comparison, right? And for this kind of comparison, people usually use one of their favorite gene. And based on just a result from one favorite housekeeping or target gene, they kind of come to conclusion whether this new cDNA kit is good or not for their application. Now, whether just looking at one or two gene is good enough or not, I'm not sure. But what we did here, we compared how this Ultra Super Mix performed with one of our older product and one of the really other high quality product available in the market. So we used a 96 gene panel. So what we did basically, we started with 1 microgram universal human reference RNA, and then we generated cDNA either with our legacy product, QScript cDNA Supermix, one of the first cDNA Supermix product launched in the market, We also generated cDNA from QScript Ultra Supermix. Remember, this was done 55 degree just in 10 minutes. And then another product launched in recent years and demand to be a high quality product from a supplier. And then after cDNA synthesis, we diluted everything to make it kind of 10 nanogram equivalent cDNA as input. And we amplified those cDNA using CyberGreen-based qPCR method with 96 gene panel. And the 96 gene panel, we decided to use here content different kind of GC into the target, as well as the expression levels are wide. So you can get one gene highly expressed and with CQ value 18 to 20. And there are also genes which has CQ value around 30, 32. So we covered wide range of GC content and wide range of expression level as well. And then for analysis, we calculated minus delta CQ by taking the CQ coming from our cDNA supermix and then subtracting the CQ coming from either Ultra Supermix or supplier A Supermix. So here is the data minus delta CQ data for 96 gene panel. And what you see is the data from our cDNA supermix is in the orange line, which is basically the baseline. And if you see the bar going up, that indicates the cDNA synthesis efficiency was better. And if anything below that orange line, that means it underperformed compared to our QScript cDNA supermix. And you can see in most cases, Ultra Supermix and the other product we use from supplier A perform better than our old cDNA supermix. But even then, you can see consistently our Ultra Supermix perform better than our old supermix and other supplier A supermix we used. So this gives us a lot of confidence. It's just not the data from one or two targets. We tested 96 panel and showed that our Ultra Supermix performed better in wide range of applications. Now, not only our data, but now different labs are using this product and a couple of publications are coming out. We are running out of time, so I'll quickly walk you through a few publications published last year or this year. So in this particular publication, they used hypothalamus, hippocampus, liver, and small intestine tissues from rat, isolated RNA using RNA-Z-kit from QIAGEN, and then used QScript Ultra Supermix for cDNA synthesis, followed by qPCR using CyberGreen-based qPCR method for relative gene expression study. So here is the data. I'm not going through their data, but here they are looking into gene expression of the control sample versus male rats having KASE. There is another publication where they're looking at the YAP pathway and they used rat alveolar epithelial cells type 2 from ATCC. They isolated RNA using nucleospin RNA plus minikit. And then for cDNA synthesis, they used QScript Ultra Supermix, followed by qPCR using Perfecta CyberGreen Fast Mix from Quantabio for relative gene expression study. Again, I'm not going into data, but they are looking at cytosolic and nuclear YAP protein expression in this particular paper. And finally, in the last example, they used transfected BHK-T7 cells, and for extraction, they used PureLink RNA mini-prep kit from Invitrogen, QScript UltraFlex kit in this particular case, where they used gene-specific primer for cDNA synthesis, and then again, cybergreen-based relative gene expression study. And here is the data where they looked at relative expression of S1 RNA level using RT-qPCR compared to housekeeping gene beta-actin. So the reason I wanted to show these three publications, you can see most cases people will use ultra supermix for cDNA for gene expression analysis. But if you ever need to use gene-specific primer, you have that option from QScript UltraFlex Kit as well. And QScript UltraFlex Kit, remember, it's kind of a master mix too. Only thing is separate is the primers. So that gives you flexibility, at the same time, reduce number of pipetting step based on the formulation we have. So with that, I want to conclude this webinar with the take-home message being QScript Ultra RT is a novel engineered reverse transcriptase. It has superior processivity. It generates greater than 20 kb cDNA just in 10 minutes reaction time. It has elevated thermostability. Optimal RT temperature is 50 to 60 degree, and Ryan mentioned that it can overcome secondary structure of RNA that can stall cDNA RT during the cDNA synthesis. And this ultra RT intrinsically have strong inhibitor resistance capability. We have two different formulations using this RT. First one is QScript UltraFlex Kit, which is an optimized system for rapid and efficient first-strand cDNA synthesis with flexibility in the choice of priming method. It is ideal for custom gene-specific primers or for generating long cDNA products. And QScript Ultra Supermix is a single tube optimized system for first strand cDNA synthesis that delivers convenience, reproducibility, and consistency, least pipetting steps, and mixed priming method. On behalf of Susan, Ryan, and the entire Quantabio team, I want to thank you for joining the webinar. And if you have any question, please post it in the Q&A session, and we'll be happy to answer it.
Thank you, Ryan. Thank you, Subrata. So we did have a couple questions come in already, so we'll go straight into it. We have just about 6 minutes to address maybe 2 or 3 questions. Whatever we don't get to today, we can reach out to you, or please reach out to us in any of the contacts that you see here. So the first one is, can Ultra RT be used in the application requiring template switching mechanism?
Yeah, so I guess I can take that one. Alternative to the MMLV class of enzymes. So the MMLV ones, again, leave that predictable non-templated addition that works well with standard template switch oligos. Whereas the QScript Ultra, it does do end addition, but it's less predictable and more random. So the standard template switch oligos, unfortunately, do not work well for template switching for this particular enzyme.
All right. And has anyone used Ultra RT for long-range sequencing in the PacBio and ONT platforms?
Yeah, so we have few laboratories working with us. They used UltraFlex Kit, and they used oligo-DT primer for longer cDNA synthesis, followed by doing generating longer amplicons using either replica hi-fi tough mix, or we have another product called Acustar long range super mix. So and then they sequenced it in PacBio platform. So very similar approach I showed during the RNA virus sequencing, but you can use oligo-DT to generate long cDNA for eukaryotic sample and then generate double-stranded DNA for sequencing as well.
And the next question is this enzyme available in a one-step PCR mix? If so, are there any advantages using this enzyme versus the one in XLT mix?
Yeah, so I mean, this is definitely something we're working on, so stay tuned for that. Compared to the mix you mentioned, one big advantage I could just think of off the top of my head is the higher verse transcription temperature capability, which allows you to get through those, difficult viral, sequences with RNA secondary structure. Yeah, and high inhibitor tolerance. Yeah, so this is definitely something we're having in the works. And yeah, again, stay tuned, and hopefully we can get back to you with more information on that soon.
Awesome. And probably the last question for today, would you share what is the secret of low duplication rate?
Yeah, so this is again something where we're kind of still investigating, but we believe that the low, the low insertion deletion mutation frequency that we found is related to the processivity. So just by staying engaged with the primer template, better, and then having that better processivity allows for less slippage, and then that sort of thing, which would lead to these insertions and deletions.
Great. Good. So again, any questions that you might have regarding the topic today or any of our Quantabio products, please reach out to us and someone will be happy to answer your questions. And thank you again, Ryan Subrata, for the great talk. And with that, we will close this webinar and the recording will be available soon and will be distributed. Thank you, everyone.
Thank you.
Thanks, everybody.